Hey Skydians, here we are with another amazing short video on the topic of Erythroblastosis Fetalis. So let's answer, what is Erythroblastosis Fetalis? Erythroblastosis Fetalis is another name for hemolytic disease of fetus and newborn, HDFN. As we said, hemolytic means in this disease there occurs abnormal lysis of the blood, as heme for blood and lysis for breakdown. So we can conclude that Erythroblastosis Fetalis is a disease in a fetus or newborn, characterized by abnormal lysis of red blood cells. This disease occurs due to the Rh incompatibility, that is, the difference between the Rh blood group of the mother and baby. Let me explain it further. We know that there are two blood group systems that are given prime importance before blood transfusion, that is, ABO system and Rh system. ABO system, based on the presence and absence of antigen A and antigen B on erythrocytes or red blood cells. And on that basis, blood is divided into four groups. Type A blood, if only antigen A is present. Type B blood, if only antigen B is present. Type AB blood, if both antigens A and B are present, but is the least common type. And the fourth type is type O blood, the most common type, where no antigen is present on erythrocytes. These antigens are inherited by the next generations, and antibodies related to each type of antigen are produced between 2 and 8 months after birth. For example, if antigen B is absent, as in type A blood group, the antibody called anti-B antibody is produced. Similarly, people with type B blood group, where antigen B is present but antigen A is absent, have anti-A antibodies. Those with type AB blood have neither of the two antibodies, whereas people with type O blood have both anti-A antibodies and anti-B antibodies. Then comes the second blood group system, that is, Rh blood group. The Rh blood group got its name from the rhesus monkey, because it was in this type of monkey the blood group was first studied. There are several Rh factors in humans, the most prevalent of which is antigen D. If present on the red blood cell membranes, the blood is called Rh positive. If not, it is called Rh negative. The presence or absence of Rh antigen is inherited, but unlike ABO blood group, the antibodies that react with it, which are called anti-Rh antibodies, are not spontaneous. They form only in Rh negative people because of specific stimulation. So, this was the basic knowledge to revise before understanding our main topic, that is, Erythroblastosis Fetalis. Let us define it properly. Erythroblastosis Fetalis is a disorder in the fetus, characterized by the presence of erythroblasts, a preformed or immature red blood cell in blood, and its main underlying cause is Rh incompatibility. When we say Rh incompatibility between mother and fetus, it means that Rh blood type of mother is different from that of the fetus she is carrying. But in the case of Erythroblastosis Fetalis, it particularly happens when mother is Rh negative, but the fetus is Rh positive. Now let's see how this happens. When a female with Rh negative blood marries a male with Rh negative, then 100% of offspring will be Rh negative. So there's no chance of Rh incompatibility. But in case if Rh negative female marries a Rh positive male, then there is a chance that 50% of their offspring are Rh negative and 50% are Rh positive. So in case when a mother is Rh negative, but the fetus is Rh positive, then there are chances of Rh incompatibility, which may lead to Erythroblastosis Fetalis. Let's understand how this incompatibility leads to this hemolytic disease. Here, we will take a closer look at the mother's blood. That is without Rh antigen, as it is Rh negative, and a fetus's blood with Rh antigen, as it is Rh positive, with a placental barrier in between. As we mentioned earlier, the anti-Rh antibody does not produce simultaneously or on its own, but is produced only when it comes in contact with Rh antigen. But it does not happen during normal pregnancy. That is the reason why usually the first child escapes the complications of Rh incompatibility. This is because the Rh antigen cannot pass from fetal blood into the mother's blood through the placental barrier. However, at the time of parturition or delivery of the child, because of placental detachment, the Rh antigen from fetal blood may leak into the mother's blood. Then during the postpartum period, that is, within a month after delivery, due to the presence of Rh antigen from the fetus, the mother develops anti-R antibodies in her blood. So now, when the mother conceives for the second time, or during the second pregnancy of that mother, and if the fetus happens to be Rh positive again, the Rh antibody from mother's blood crosses the placental barrier and enters the fetal blood. As we know, Ag can cross the placental barrier. The Rh antigen cannot cross the placental barrier, whereas Rh antibody can cross it. So, when an antibody crosses the placental barrier and enters the fetus, it reacts with the Rh antigen present on fetal red blood cells, thus causing agglutination of red blood cells, resulting in hemolysis. This severe hemolysis in the fetus causes jaundice. And now, as the number of red blood cells rapidly falls in the fetus, so to compensate this hemolysis, there is more and more rapid production of red blood cells, not only from bone marrow, but also from the spleen and liver. Now, many large and immature cells in pro-erythroblastic stage are released into circulation. Because of this, the disease is called Erythroblastosis Fetalis, means more erythroblasts in bloodstream of a fetus. Ultimately, due to excessive hemolysis, severe complications develop like excessive hemolysis results in anemia, and the infant dies when anemia becomes severe. It may also lead to Hydrops fetalis. It is a serious condition in the fetus, characterized by edema. Severe hemolysis results in the development of edema, enlargement of the liver and spleen, and even cardiac failure. When this condition becomes more severe, it may lead to intrauterine death of the fetus. And another severe complication is Kernicterus. It is the form of brain damage in infants, caused by severe jaundice due to hemolysis. If the baby survives anemia in erythroblastosis fetalis, then Kernicterus develops because of high bilirubin content. The bilirubin enters the brain and causes permanent brain damage. So what could be the possible preventions and treatment of erythroblastosis fetalis? Well, if the mother is found to be Rh negative and fetus is Rh positive, antibody against the antigen or anti-Rh antibody should be administered to the mother at 28th and 34th weeks of gestation as prophylactic measure. If Rh negative mother delivers Rh positive baby, then anti-D should be administered to the mother within 48 hours of delivery. This develops passive immunity and prevents the natural formation of Rh antibodies in the mother's blood, thus no long-term active immunity. So, the hemolytic disease of newborns does not occur in a subsequent pregnancy. And second, if the baby is born with erythroblastosis fetalis, the treatment is given by means of exchange blood transfusion. Rh negative blood is transfused into the infant, replacing infant's own Rh positive blood. It will now take at least 6 months for the infant's new Rh positive blood to replace the transfused Rh negative blood. By this time, all the molecules of Rh antibody derived from the mother get destroyed. So this was all about hemolytic disease of fetus and newborn. that is named Erythroblastosis Fetalis. For more such interesting videos, keep visiting sqadia.com.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.